Hello and welcome back to SCI Teach. This is a chemistry practical salt analysis lesson for NECO 2024. So we're going to analyze the salts from the specimens, the salt C, and also analyze D. We're going to analyze and see the ions that are present in the salt C and D. We're going to do a full analysis. The questions in your exam may not cover the whole analysis, but we're doing a full analysis so that wherever the question come out from, you can answer them appropriately. All right, so let's go. Now I made a table of test, observation, and inference of the salts C and D. The salt C is what we're going to analyze first, and then we're going to test for the ions present in C and also in D. So I'm going to take you through the table step by step, and when we finish a particular test for a particular ion, then I'll show you practically how it's done. Then we move to the next ion and so on and so forth. So we start with number 1A in your table there. As we take the salt that is labeled C, we add water. The examiner may tell you to add about 10 cm cube of water, whatever it's that you are asked to do in the instructions. The idea there is that C plus water, you added water to it. Then he will ask you to stir it so that the salt will dissolve completely or whatever. And then you divide the solution into 2 portions. This is my own thinking now because of the salt I want to test. Yours may not be divided because you may not be asked to test both Cl and zinc ion. But if you're asked to, possibly they may ask you to divide the solution into 2 portions, or they just say when you add water to C and then you stir, and then you take 2 cm cube of the solution of C into another test tube and carry out the practical. But I'm putting my own this way so understand. So when I do that, C plus water, and I stir that, and I will discover a clear and colorless solution formed. So I infer that C is a soluble salt. Now since it forms a clear and colorless solution, then it's soluble, it dissolved completely. Then B, the I of B, for the first portion of C, add dilute trioxynitrate 5 acid, HNO3. And if you add HNO3 to this portion of C, you will discover that there is no visible reaction. Nothing happens, as if you're just adding water to water. That's how it will look like. So you observe that there is no visible reaction. As there is no color change, there is no precipitate formed, there is no effervescence, so no visible reaction. But then you add silver nitrate, AgNO3, to the mixture of C and HNO3 that you had no visible reaction. Add silver nitrate to that mixture, and then you'll see a white precipitate formed. And when this white precipitate forms, that is a positive test for chloride ions. Okay, what it means is that C must have contained chloride ions. And so when silver nitrate, AgNO3, was added to that solution, AgNO3 saw a chloride there, and Ag bonded with the Cl ions and formed AgCl, silver chloride. And silver chloride is forms the white precipitate. So that's why the white precipitate is formed. So that's to show that Ag has formed a precipitate or formed a compound with chlorine, which is silver chloride, and silver chloride is what formed the white precipitate. So it means that there is chloride ion in the solution. So you say Cl minus present. But let's go ahead and try and confirm if that was really Cl minus. So II, the mixture in Bi, that's the mixture of C and HNO3 and AgNO3, that mixture, add excess ammonia to that mixture. If you do that, you will discover that that white precipitate that was formed when silver nitrate was added, that's that white precipitate of silver chloride, will dissolve when you add excess ammonia. And this is a clear confirmation of chloride ion. So we infer that Cl minus chloride ion is confirmed. All right, so let's see how that is carried out. So here is the practical. So we add the salt that contains the chloride ion into a test tube. You know, this is C. We add C into a test tube, and then we add water. Remember, it was C plus water. So we add C to what? Now C can be we from our own view there was contain chloride ions, and then so it could be any sort of that contain chloride. So but you just put the C in water and stir. And to the first portion, remember, we are asked to add dilute HNO3. So we add dilute HNO3 to it. Okay, about that. And then we're then told, we see there's no visible reaction. You look at it, you see it's a clear solution, nothing happened. All right, see clearly. Now we are then asked to add silver nitrate, but I didn't have silver nitrate, so I'm using lead nitrate. Using lead nitrate here, it's going to still form this time lead chloride. So as we add it, you see a white precipitate is formed. This white precipitate, in this case, since I use lead nitrate, this white precipitate is lead chloride. Okay, in your own, you'll be asked to use silver nitrate, which the white precipitate will be silver chloride, but this is lead chloride. Now that's confirmed that there is chloride ions in the solution. But we're going to because I'm using lead nitrate, I can't add excess ammonia. Excess ammonia, it will not dissolve. It will not, the precipitate will not dissolve in excess ammonia. But if I used silver nitrate, it would dissolve. Okay, but if we were to use lead nitrate and you want to still confirm, we would need to warm the solution, heat the solution, just put it on over a source of heat, and then heat it. When you warm it, you see that the precipitate will dissolve on warming, but if you allow it to stand, it will precipitate will come back, and that will be a confirmation that there are chloride ions in the solution. So but if as your case would be in your exam, silver nitrate, you go ahead and do II, which is add excess ammonia to the solution that formed that white precipitate, and when you add the ammonia, you will discover that the precipitate will dissolve, and you confirm that Cl minus is present. So we've tested for chloride ions, and we want to test for zinc ions. So go back to our table and go to 1C, and remember first of all that 1A, we said C plus water, and then we stirred and divided the solution into two portions. The first portion, we used it to test for Cl minus ions. Now the second portion of this C solution is what we are using now. So to the second portion of C, we add ammonia, NH3, in drops. If we add ammonia in drops, we'll discover that a white gelatinous precipitate will be formed, a jelly like sticky white in color precipitate will be formed. And when this is formed, we'll suspect that this could be zinc Zn2 plus or aluminum Al3 plus ions that are present. So we infer that since we observe that the white gelatinous precipitate is formed when it reacted with ammonia, then it may either be zinc ion or aluminum ion. But let's confirm which one it is, because it can't be both of them. So we add this ammonia now in excess. So we put it in excess, not in drops anymore. And when we add it in excess, we discover that the precipitate will dissolve. And with the precipitate dissolves, then it means we are sure now that, all right, it wasn't aluminum that was present, it is actually zinc ion that is present. So we infer Zn2 plus confirmed that zinc ions are confirmed present in that solution of C. That would mean that the salt C, since it contains chlorine, chloride ions, and zinc ion, then that means C was zinc chloride salt. So let's see how it's carried out. Now we are testing for zinc ions. All right, remember that zinc ions can be tested using sodium hydroxide and NH3. So this video I'm going to show you how to use it both ways. Okay, sodium hydroxide and then NH3, but NH3 is what is in our table. So we dissolve a salt that contains the zinc ions into solution and mix it properly. Okay, so, and since we're going to test with sodium hydroxide and NH3, we divide the mixture of the solution into two parts. So the first part we're going to test with sodium hydroxide. So we'll add sodium hydroxide in drops into this solution, and if we add sodium hydroxide in drops, you will see that a white precipitate will be formed. A white precipitate will be formed. You can see that clearly, and it's gelatinous. So a white gelatinous precipitate will be formed. See how it is, as though it sticks to the walls. All right, so a white precipitate is formed. If you add sodium hydroxide in excess, you discover that the white precipitate does not dissolve. Okay, see clearly, the white precipitate will not dissolve, and even when you shake to mix it or to try and force it to dissolve, it does not dissolve. So zinc ions does not dissolve in excess sodium hydroxide. But the one in our table here is the test with ammonia, NH3. So we take the other portion of the solution of salt that contains the zinc ion, and then we react with ammonia. Remember our table says ammonia in drops first. Okay, so if we add ammonia in drops, you see the white gelatinous precipitate formed also. So you can see it clearly now that the white gelatinous precipitate is formed. You see, sticks to the wall, so it's gelatinous, as gummy or paps like. So a white gelatinous precipitate is formed, but the difference between this ammonia and sodium hydroxide is that if you add excess of ammonia to the white precipitate solution, the white precipitate will dissolve. So ammonia dissolves in zinc ions dissolves in excess ammonia. So the precipitate that forms when you add ammonia in drops dissolves when you add ammonia in excess. That's what is in our table. The white gelatinous precipitate is formed, and the precipitate dissolves when you add ammonia in excess, and that's a confirmation that Zn2 plus is in that solution, not Al3 plus like we suspected that zinc or aluminum may be present. So lastly, we will test for the last last substance. That's D. We've tested for C, and we found that C contains chloride ions and zinc ions. That means C is zinc chloride salt. Okay, now D is another substance now, and we'll be asked, put this D in water, stir it, and to me, divide it into 2 portions. All right. Now, if you put D in water and you stir it, you will see a white or creamy solution with colloids inside. See particles inside that does not want to dissolve, but they are uniformly spread in the solution, and but the solution itself is creamy in color, or white, some sort of milky color. And so you observe that and write a white or creamy or cream colloidal solution formed. And what that means is that D is a heterogeneous mixture. Heterogeneous mixtures don't combine uniformly. They don't mix up in such a way that you don't see the difference between the 2 or more components. But this one, you see the particles inside. They are not totally mixed up together with the water and form a homogeneous mixture. So it's heterogeneous mixture. So we then go ahead and divide into 2 portions, and then to the first portion of the D we add copper sulfate. When we add copper sulfate, we'll discover that the blue color of copper sulfate will become deeper, and it will deepen. So I observe that and I write it, that the blue color of CuSO4 deepens. And then I go ahead to that mixture of D and CuSO4, I add sodium hydroxide to it, and I discover that the solution changes in color from that blue color to purple color. Now that is Burette test. Burette test is a test for protein. So the reagent in Burette test is copper sulfate and sodium hydroxide. If you add copper sulfate to a substance, a solution that contains protein, then the copper sulfate, when sodium hydroxide is added to the solution of copper sulfate and the substance that contains the protein, a purple color will be observed. In fact, for the sake of question 3, which may want to ask question more on the Burette test and all those, which I suspect, Burette test is just a chemical test that tests for peptide bonds, and peptide bonds are the bonds that hold amino acids together, so that the amino acids will come together to make up proteins. So indirectly, Burette test is used to measure the amount of protein in a substance. The reaction will lead to a color change to purple, and then the deepness of the purple will depend on the amount of protein in the sample. If the protein is much in that sample, then the purple color will be deep, very deep. But if it's less, the purple color will be a little lighter. So the deepness of the purple depends on the amount of protein in the sample. All right. Then in alkaline solution, that's if you add an alkali like sodium hydroxide, a base, to the Burette's, in alkaline solution, the copper 2 will form a complex. That's a peptide copper complex. It forms a complex with the peptide bonds. Okay, so the peptide will bond with the copper and form a complex. So peptide copper complex, something like that. Once this complex is formed, the solution will turn from the blue color of copper 2 to a purple color. All right. That is the Burette test, in case you are asked. So that's what we just did here, and we added CuSO4 copper sulfate to the first portion of D, and then we saw that the blue color of CuSO4 was there and just a little deeper. And then when we added sodium hydroxide, an alkali, remember in alkaline solution, the copper 2 in CuSO4 formed this complex with the peptide bond, and then because of this complex that is formed, the solution turned from blue to purple. And so when that happens, we then say, okay, so that means protein is present. That means that solution of D was protein. But let's go ahead and confirm if that was really protein. So we test with something else, with trioxonitrate 5 acid, yes, HNO3. So to the second portion of D, we add HNO3. And when we add it, we'll discover that a yellow color will be formed. A yellow color will be observed, a persistent yellow color. So the solution that was whitish or creamy will turn to yellow. And when that turns to yellow, then you've confirmed that protein is in that substance. So protein confirmed. So let's see how that is carried out. So for the last one, D, we were asked to put, as 2A, we were asked to put D in water. So D plus water, and then we stir it. So you can see that a white or creamy colloidal solution is formed. Now we're going to divide the solution into 2 portions, but we'll just take a little out of it, use as the first portion. All right. Now we are going to add to the first portion copper sulfate. You see the blue color of copper sulfate now. So we're going to add this copper sulfate to this solution, and you can see the blue color of copper sulfate inside the solution. You can see it clearly that the blue color of copper sulfate is present there. Okay, now we are going to add sodium hydroxide to the solution and see what happens. If we add sodium hydroxide, you see that the color of the blue is changing. All right, you see clearly the color of the blue color of the copper 2 ions is changing. Now don't end your practical here. You see this color is still color of copper you are seeing here, because the copper is still more than the sodium hydroxide right at this point. So don't stop here. Remember, we want to see a purple color, so you keep adding until you see that. Also depends on the concentration of your sodium hydroxide also. So you see that the color is turned purple now. Now the salt or the solution changes from blue to purple, so you see clearly that we have a purple color. Remember, we said that the deepness of the purple depends on the amount of protein found in the solution. So you see how deep our purple is. Now let's confirm if it's really protein that is there. So we add a little amount of the D solution into another test tube, and then this time around we're going to test with nitric acid, that's trioxonitrate 5 acid. So if we add trioxonitrate 5 acid to the portion of D, we see that we have a persistent yellow color. So we see the yellow color formed, and this is a confirmation that protein is present in that solution. So you see the yellow color that's formed here. If you want, you can compare. You see that the one that used Biret test formed a purple color, which is on the right hand side now, and the one you used HNO3 formed this yellow color. So these are different results you get from the test for protein. All right, so that's the end of the salt analysis for today. Please make sure you go through it over and over again and understand better. If you have questions, drop them in the comment section and I will answer your questions. Remember also to subscribe to this channel so that you have access to more enriching contents. Also share this video to your friends and loved ones that have exams to write, so that they will also benefit from something good. Thank you very much. God bless you.